So overall, pneumotaxic center, if it is strong, it will inhibit the apneustic center. If it is weak, it will stimulate the apneustic center. The apneustic center, it will act on DRG in quiet breathing, and in forceful breathing it will act on VRG. DRG and VRG, they will work in reciprocal inhibition pattern. So apneustic center, if it is stimulated for longer time, it will generate long inspiration with short expiration. So apneustic center will have stimulatory effect on DRG, and if it is inhibited, it will have inhibitory effect on DRG. So if the inspiration is needed much stronger, so it will have inspiratory neurons of VRG also enrolled, and in forceful expiration it will act on the expiratory neurons of VRG. Okay, so this is how there is the overall regulation of respiration. Now this DRG is getting in many afferents. So one of the important reflex, that is Herring-Brewer reflex. I will talk about this Herring-Brewer reflex. So this reflex is important because it has two parts. One is inflation, and there is deflation. Okay, so we'll talk about inflation reflex. Opposite to this will be deflation reflex. So what happens in this? The stretch receptors which are present in the muscle wall of bronchi and bronchioles, the muscles of bronchioles and bronchi, they are stimulated if the inspiration is increased. So usually if the inspiration is increased than more than 3 times the normal tidal volume, means if it is more than or approximately equal to 1.5 liters per breath, these stretch receptors, they are stimulated. These stretch receptors, they will carry information via 10th nerve to DRG, and in DRG they will act on switch off point. They will act on switch off point, and they will stop the ramp signal, which will stop further inflation. Okay, so basically this stretch receptors in Herring-Brewer reflex, inflation reflex, will act as a pneumotaxic center. Okay, a strong pneumotaxic center which is halting the inspiration by stopping a ramp signal by controlling the switch off point when the inspiration is more than 3 times the tidal volume, that is 1.5 liters per breath. So this is called as Herring-Brewer's inflation reflex. Opposite to that is deflection reflex. Okay, so it will prevent the overstretching of lungs, and it will cause stoppage of inspiration, further inspiration. So next we will talk about chemical regulation. So in chemical regulation of respiration, it is done by central chemoreceptors and peripheral chemoreceptors. So peripheral receptors are further carotid and aortic chemoreceptors. Okay, central chemoreceptor is present in the ventral medulla. It is very sensitive to changes in PCO2. There is rise in PCO2, it will cause increased in ventilation. So now how it is occurring, we'll see in details. The carotid and aortic, they are sensitive for decrease PO2 and changes in pH. So basically the chemical regulation is done, or it occurs when there is altered chemical concentration of CO2, oxygen and pH. So main function of the lung is to maintain the carbon dioxide and oxygen partial pressure inside the pulmonary circulation. So if the PCO2 is high, if there is decrease in PO2, and if the pH, that is increased in hydrogen ion concentration, the pH is low, there will be stimulation of chemoreceptors and it will cause increased ventilation to counteract this part. So basically there will be increase in respiratory rate. So if there is more PCO2, the respiratory rate will increase and there will be more CO2 washout. If there is low oxygen partial pressure, the respiratory rate will increase and there will be more oxygenation of alveoli. If the hydrogen ion concentration is more by buffer system, there will be increase in respiratory rate and the pH is maintained. Okay. So now how there is regulation of respiration by central chemoreceptors. So central chemoreceptor is sensitive for PCO2 increased and decreased pH, that is increased hydrogen ion concentration and decreased PO2 too. So most important stimulant is increase in PCO2. Now if there is increase in partial pressure of carbon dioxide, this carbon dioxide, it cannot cross blood-brain barrier. Okay, so it cannot cross blood-brain barrier, but what it can do is PCO2 will combine with water and it will form carbonic acid, which will split up into hydrogen ion and bicarbonate. Okay. So this hydrogen ion, it can cross blood-brain barrier. So the central chemoreceptor, which is present in the ventral medulla, it will sense the increase in hydrogen ion concentration, which is because of change in PCO2. So this is called as indirect chemical regulation. So indirect conversion of carbon dioxide into hydrogen ion concentrations, which is done in the blood-brain barrier, which can cross the blood-brain barrier, and the CSF, which is present inside the fourth ventricle in the medulla, it will sense the change in the hydrogen ion concentration and it will cause direct stimulation of respiratory center. Direct stimulation of respiratory center, that is DRG, leading to increase in respiratory rate, which will cause increase in CO2 washout. So this will cause bringing back the PCO2 to normal. Okay. So the central chemoreceptor, they will act by increasing PCO2 via this mechanism. But this will remain for acute period. Okay. Because after 1 or 2 days, the renal buffer system will get activated. Renal buffer system will take over, and this hydrogen ion is, which is released by increased PCO2, will be handled by renal increased bicarbonate reabsorption. Okay. So in renal system, there will be increase in reabsorption of bicarbonate, which will combine with this hydrogen ions and it will form carbonic acid, and so there will be less hydrogen ion concentration in the cerebral spinal fluid and there will be decreased or coming back of respiratory rate. Okay. So this is for acute condition. If there is increased PCO2, the chemoreceptor, central chemoreceptor will act. Okay. after 1 or 2 days the renal buffer system will take care of hydrogen ion concentration. Now next is peripheral chemoreceptors. So when we talk about peripheral chemoreceptors, first carotid. The carotid body, which is present in the bifurcation of carotid artery, at the bifurcation of carotid artery, this carotid body is very sensitive for change in the PO2. So if there is decrease in PO2, because it is getting 20 times more blood supply than any other organ. So a slight change in PO2 will cause stimulation of this peripheral chemoreceptor, that is carotid body. How they are acting? There are two types of cells, type 1 and type 2 cells. The glomus cells, type 2 cells, they are having afferents. Now these afferents will go via 9th cranial nerve to DRG. Now how they are activated? They will cause decrease in potassium efflux by reducing the conductance of potassium channel. Okay, so conductance of potassium is reduced, so decrease in potassium efflux, which causes depolarization of nerve, which will increase the release of neurotransmitters, mainly D2 receptors, neurotransmitters, which will generate the action potential, and it will be carried to respiratory center, and there will be stimulation of DRG, which will cause increase in ventilation, so that the PO2 will come back to normal. So this is how there is activation of carotid receptors via 9th nerve. Same way the aortic body, they will also sense the change in PO2. They will carry this information to the respiratory center, that is DRG. Now this is via 10th nerve. Okay, 10th nerve, it will carry the information to DRG. Here also same way there will be decrease in potassium efflux. So because of decreased cyclic AMP, decreased potassium efflux, there is opening of calcium channels, release of neurotransmitters, then production of action potential, and then it is carried out to the respiratory center DRG and increase in ventilation. So this is by peripheral chemoreceptor, that is aortic body. So in chemical regulation, the change in PCO2, PO2, pH and ion. So basically central chemoreceptors, they are activated by PCO2, peripheral they are activated by PO2. The primary chemical change is this, and the secondary by PO2 and pH. Decrease PO2 and decrease pH. Same way in peripheral also, PCO2 if it is raised, this peripheral chemoreceptors will get activated, and if the pH is low, this peripheral chemoreceptors will get activated. So this is how there is regulation of respiration by chemical mechanism. So basically all these altered changes, it should come to normal. So if you see in statography, if you voluntarily hold the breath, there will be CO2 accumulation. After that voluntary breath holding, if you release that point, there will be long inspiration. Now this long inspiration is because of CO2 accumulation causing stimulatory effect on respiratory center. So this will cause increase in CO2 washout. Okay, this is during voluntary breath holding. Okay, then when you are doing exercise,